Okay, so we will start in 1 minute. So we have almost everybody in already. We will start slowly. Julianne, maybe you can share your presentation and get ready.
Hello, welcome everyone. I will start sharing my screen. Excuse me. Hello, welcome everybody. Welcome to this webinar. Today we are very excited to introduce to you Stratus, our cloud-based bioinformatics platform for automated and seamless analysis of NGS data. My name is Julien Lagarde. I'm head of bioinformatics at Flomics. And together with Cristina Atugni, bioinformatics scientist at Flomics, I will present Stratus to you today. First things first, who are we? Who is Flomics and what do we do? So Flomics was founded in 2018 at the PRBB. We've now moved to the Cosimbio co-working space building at Poblenou in Barcelona. And our core project is to develop a test based on liquid biopsy and cell-free RNA-seq for the early detection of different types of cancer. Scientifically, we're specialized in next-generation sequencing, molecular oncology, metagenomics, RNA-seq analysis, etc. Apart from this core project and expertise, we also provide genomics and bioinformatics services to customers, both in the wet and in the dry lab. So this is what we like to call the Flomics wheel of expertise, and this summarizes our know-how. What you can see on the left-hand side of this wheel is our wet lab expertise, so to say. So first, on the bottom left, you can see our core project, liquid biopsy solution, which is in development for the screening monitoring of cancer. We also specialize in next generation sequencing, as I said earlier, and we provide genomics services for the wet lab. So we can do wet lab experiments for you. And if you're interested in this aspect of our research and services, I invite you to attend this free webinar on the 15th of February, given by our colleagues Yuk Cabuz and Mercedes Serrano. You can register for free using the provided links at the bottom. Today, the topic of our webinar is bioinformatics, as the title and description of the webinar say. And this is subdivided in two services at Flomics. So we provide custom bioinformatics services. This is where our bioinformaticians run custom analysis for you. And for independent autonomous bioinformatics analysis, we provide Stratus, our NGS cloud analysis software. So as a background, this is what an NGS analysis workflow looks like. Typically, this starts in the wet lab with quality control, library preparation, etc. Then followed by sequencing of those libraries. And sequencing is about basically turning nucleic acids into big data, basically FASTQ files. So we can do this at Flomics for you as a service. And we can also do the second part or the third part of this workflow, which is bioinformatics, which is what we're focusing on here. And bioinformatics is basically about understanding, interpreting this big data. Bioinformatics analysis can be subdivided into primary, secondary, tertiary analysis based on the level of sophistication. Primary analysis is basic quality control of your data, followed by read alignment, and quality control at each step, all the way to a final interpretable report and possibly published papers. So the trick here is that not everybody can do that, because this requires bioinformatics skills and also a strong computing infrastructure. So to give you an example, if you want to map RNA-seq reads to the genome, to the human genome, you will need a computer of at least 40 gigs of RAM, which not everybody can afford. So on top of these skills, you need a strong infrastructure. And this is what we propose at Flomics. So we have a first-class teams full of incredible talents, as you can see here. So we have data analysts, bioinformaticians, as well as talented software developers. This is our team. And in terms of infrastructure, we use a state-of-the-art computing infrastructure. We're 100% on the cloud. We're using Amazon Web Services. And this means we are fully scalable. And we have a virtually unlimited computing capacity, both in terms of CPU, memory, or storage. We use software, excuse me, software frameworks such as Docker and Nextflow that enable full traceability and reproducibility of our pipeline. So we take these things very, very seriously, both for custom analysis and for the Stratus Cloud platform. So now moving on to Stratus Cloud platform. So this is, as I said earlier, for autonomous bioinformatics analysis, and this is how it works. So this is the homepage listing the available pipelines currently on Stratus. You can access it using this URL below. And we have currently several pipelines available for you, specialized in metagenomics, both 16S amplicon and shotgun. We have also viral metagenomics analysis pipelines, such as SARS-CoV-2 clinical for swabs, typically in the clinic, and SARS-CoV-2 wastewaters analysis for disease monitoring on the larger scale. These two pipelines on the bottom are being repurposed for any kind of virus or pandemic. Excuse me. All these pipelines follow what we like to call the four-step bioinformatics analysis paradigm. Basically, this is about selecting a pipeline in the first step, providing request details about your experiments so that we run the proper pipeline with the proper parameter, load FASTQ files. Typically, all these pipelines take FASTQ files as input, and then pressing submit. This is very easy. Now I will show you just an example with the 16S amplicon-seq metagenomics analysis pipeline. So this is basically what it does. So you will get, you provide your FASTQ files for environmental or metagenomics samples, run through an amplicon-seq protocol. And the output will be an automated bacterial metagenomics analysis, extended data quality control, which also is provided by any pipeline on Stratus, phylogenetic classification, cross-sample aggregated or comparative analysis, alpha-beta diversity analysis, etc. I don't want to spoil anything, because we'll have a live demo in a minute by Christina on this pipeline. You may have noticed on this screenshot that all pipelines have the select button, which means that they can be used currently, except this one, the DNA VAR and RNA-seq. This is because they are still under active development, but will be soon active. So DNA VAR allows you to detect somatic variants in any kind of sample and provide clinical interpretation, variant interpretation. The RNA-seq pipeline, as we intended to be, will provide a complete RNA sequencing analysis pipeline from extensive quality control to gene and transcript quantification. Moving on to custom bioinformatics services. So as I said just now, the DNA variation and RNA-seq pipelines are not ready on Stratus yet, but what we can do for you, if you're interested in this kind of analysis, is run it, perform it as a custom bioinformatics service run by our bioinformatics team at Flomics. This is an example of a custom RNA-seq analysis that we performed for a customer recently. In that case, we performed even the wet lab experiments, library preparation, etc. This was in mouse. And we performed basic quality control, extensive quality control, differential gene expression analysis. We provided tracks to be loaded on Ensemble or other genome browsers, as well as pathway KEGG analysis. On DNA variant calling and interpretation, we can provide a full end-to-end pipeline for you, including the wet lab. So this can go from sample collection to variant clinical reports, going through sample collection, panel sequencing, bioinformatics analysis, quality control, and all the way to clinical reports. So in this case, we go through bioinformatics analysis, but we go one step further into clinical interpretation. So if we work in the clinic as an oncologist, for example, this can be very useful for you. This is an example of a report we can provide for a patient. This is based on a excuse me, on a liquid biopsy from a patient, and we provide a summary of somatic mutations detected in the sample, all the way to clinical therapy guidance. Another typical request we get from other scientists has to do with biomarker discovery, and typically this is a scientist coming to us saying, hey, I've discovered this and this biomarker in a tumor sample, for example, and I would like to know if it has potential as a biomarker in plasma. So what we can do is, given a list of such potential biomarkers, we can scan a database of several thousand cell-free RNA-seq samples from plasma, both generated in-house and externally. And we can report to you different detection levels of these specific genes in plasma across different phenotypes, healthy or disease, for example. And this is typically very useful for assessing the potential of gene biomarkers for liquid biopsies in plasma. So this is just two examples of the kind of genomics and bioinformatics services that we can provide. We are open to all custom analysis requests, resources permitting, of course, and we're basically interested in contributing to any exciting scientific project. Sorry. So now I will pass the screen and the microphone to Christina for the live demo of the metagenomics amplicon-seq pipeline. Don't forget to write your questions, if you have any, in the dedicated channel. Thank you. Christina, you can take over now.
Thank you. Thank you, Julien. So welcome to Stratus. This is the login screen of our Stratus platform. Using Stratus is as easy as creating an account. To register an account, users need to input password and their email, and all of their data is protected behind the password. And we also make sure to be compliant with all of the data protection regulations. So I'll log in to my Stratus account. And the first thing I see is this dashboard where I can see an overview of all the analysis, all of the requests are done, with a summary here of my total request, ordered by latest activity, and some insights on the amount of analysis I've been submitting per day. So Stratus is run by credits. If I have credits, I can submit samples to the several pipelines we have, but here on the top right screen, you can see that now my account has 0 credits. So first thing should be to fund my account. By clicking fund my account, I can redeem the voucher code that will give me credits. If I did not have this voucher code, I should click here on the buy credits and fill this form, and Flomics team would get in touch with the user, with you, providing a voucher code. So I'll find my account. And now you can see that the voucher has been redeemed and I have 10 credits, which is enough to submit my analysis now. So by clicking on submit request, I'm presented with the pipeline selection screen. Let's say that I have some samples that I've prepared following a 16S metagenomics protocol. I have received the raw FASTQ files, and now I want to analyze it. I would select this pipeline. Stratus has also an implementation with the Illumina BaaS website, which means that if I authenticate and I have my samples on Illumina BaaS, Stratus takes care of directly downloading the FASTQ files without me having to download them locally on my machine and then upload them to Stratus. By selecting the 16S pipeline, now I'm presented with the request details screen, where I can select several parameters regarding how I prepared those samples. If I want more information on any of those fields, I would click here on the question mark, and this would take me to the pipeline manual, where all of the options are explained. So first, I would select the sequencing kit. For all Stratus pipelines, we try to make sure that they have the most widely used kits and protocols, but if users can't find their preferred platform kit, etc., we also make sure to cater to users, and it's as easy as getting in contact with us, and we'll do what we can to include the kits in the pipeline. So I would select the parameters that fit the samples I have. Those samples were prepared by an Illumina platform. I have the raw data, so it has not been trimmed yet. And I want to map those AmpliSec 16S samples to this taxonomy database. And finally, I need to select if the samples are single-end or per-end. And in my case, they are per-end. And by clicking next, I can see a summary of the parameters I chose. And I can choose whether if I want to upload samples from my computer or from Illumina BaaS. If I select Illumina BaaS, I can select the file that I authorized Stratus to read from my BaaS, but I have the samples on my laptop, so I would upload them. And here I have all of the samples. So I have samples from metagenome, from gut microbiome, from the colon, the jejunum, and the ileum. And I have replicates of them. Using this group column, I can select how I want to group them for alpha and beta diversity analysis. Since I want to see the difference in the metagenome distribution between the colon, mesogenome, and the ileum, the grouping should be the sampling site of those samples. But if I had treated those samples differently with two antibiotics, let's say, I would group them with antibiotic A and antibiotic B. I can also change the name of the samples, but since the sample names already fit my research question, I leave them like they are, and then I would just click next. And the files would upload to Stratus, and now I would just need to wait for the analysis to complete. As Julian has said, the computation infrastructure behind Stratus is virtually scalable as we want. So the computation time is not dependent on the number of samples, but on the sequencing depth of them, and in this case, the database used. So it would take the same for me to analyze 6 samples as to analyze 90 samples, let's say. But instead of waiting for this request to complete, I would go to the analysis results page where I already have some requests completed. You can see that the request that we just launched is here, has the ongoing status. Here I have all of my personal requests. By clicking this checkbox and selecting the actions box, I can create a shareable link for the request. And by clicking this icon, I can copy this results link and share them with whomever I want. If I want to delete that link to make this request available only for me, I would just delete the shared link. Once Stratus requests complete, I do not need to be attentive to the platform. I just can wait to receive an email to my Stratus account where I'm notified that the request has been completed. I can click on the View Reports button here, and this button would take me to this same request. So if we click on the request, we have this pop-up where I have all of the results that have been produced by the pipeline. I first have a quick quality control report. I have an aggregated report, including insights on all of the samples together. For this pipeline, this is important for alpha and beta diversity analysis. And finally, there's this button, which is a zip file that contains all of the intermediate results and reports that the pipeline has produced. So users can download everything and do their own downstream analysis if they wish. And also, for transparency's sake, users are in charge of their data. All of the reports here are produced using the contents of this zip file. And we also have individual sample reports. So I would show an example report, this one for the sample. All of our reports are interactive. You can click on several plots and zoom in or out of them. For this report, you have some summary of the run parameters if users would like to reproduce these results. Some QC statistics for this pipeline. It's regarding the number of input and trim read pairs. And in this case, the alpha rarefaction curve for metagenomic studies. This is a curve that speaks on whether the biological diversity of the sample was correctly captured with the sequencing depth that was using. In this case, if the curve reaches a plateau, which it does, it means that we have captured all of the diversity. And as I said, plots are interactive. You can zoom in if you want more granularity. You can hover over to get information on each point, and you can export this plot into a PNG to share it, etc. Then for this specific pipeline, of course, the interesting thing, it's the taxonomy assigned. We see that for this sample, 402 ASVs, that this stands for amplicon sequencing variant, were found. This means that there were 402 species in this sample, different species, so to say. But of course, we're interested in the relative abundance of those, and we present it with two plots. This first one, the Krona plot, which gives a quick overview on the diversity of the sample. Again, this is an interactive plot, so I can zoom in to a trace and then get more insights on it. And also with these bar plots. We present these bar plots for the several taxonomic levels that the pipeline is able to assign. And we can get granularity up until the genus and species. As I said, all of these plots are interactive. If I want to know what trace is this, I can zoom here and I have more resolution. And the tables are also interactive. If I want to know the frequency of a specific bacteria, I can use the search box, and I would be presented with relative frequency in this sample. And of course, you have the help and contact buttons to get further assistance. Then I present the aggregated analysis report, where we also are shown insights on these samples. If I wanted to draw conclusions on this analysis, maybe, for example, I could go to the genus and species and see that for the samples that were sampled from the ileum, there's a big dysbiosis in one of them, and this bar belongs to this species, the Clostridia badin BB60. Again, all of these plots are interactive, tables are interactive as well. So these samples come from a study where they are studying the dysbiosis happening in these three anatomical sites after inducing an inflammatory syndrome. But the advantage of Stratus is that the researchers for the study had to do all of the bioinformatic analysis manually and step by step. I just needed to upload the samples to Stratus and wait a bit to get an email that notified me that I had all of these reports ready. Then there's some alpha diversity analysis insights in the form of a box plot. This speaks on intra-sample variability. Another insight we can get is that in general, the column samples have more diversity than the ileum samples. And the beta diversity analysis. How do the samples cluster together in terms of microbiome diversity? As it is expected, despite the dysbiosis in the ileum, the two column samples cluster together, two ileum samples do as well, and the two jejunum samples also cluster together. And finally, the last plot that this pipeline produces is this sample clustering heat map. This is a heat map that it's produced using the top 30 most abundant features or species, let's say, across samples. And it allows you to compare which species are present or absent and in which abundance between samples and between groups of samples. So now that I've done this, and I've done it very easily, another thing that I can do is that everything that I've done, I can do it in an organization manner. Stratus has the organizations feature when I can aggregate users to my organization, and I can assign them credits. For example, now I have 10 credits, but I could transfer all of my credits to the organization and give credits to the other users. And I would also be able to see their requests. If I click this Share Credits button, I do not need to assign credits to users. Users all share the credits available for the organization. And finally, last important feature on Stratus is that we have this support center where we include this extensive user manual that in reality goes over everything I covered on this demo. And we also have the ticketing system. If users have some doubts, some problems, some questions, it's as easy as creating a ticket. Our support team can answer to these tickets using a Stratus platform, and we are also available and responsive to users. And that marks the end of the demo. And now we would love to answer all of your questions. Thank you.
Thank you, Cristina. So, as we said in the beginning, we invited everybody to write questions here in the chat section of this platform. I will just select a few of them and ask. So there's still time to write your questions. If you have any question for our team, just put it up there. If we don't have time, then we will address them individually and we'll get back to you, okay? So I will start slowly. And first of all, so Cristina, Julian, are there any plans to support ONT as a sequencing platform?
So I think I can answer that. So far with our pipelines, the read quality that's obtained from ONT platform is not high enough for our standards, but this could be a future project, of course.
Okay, thank you. And in terms of primers, how is it possible to customize the list of primers that are supported by the platform?
So I would say that it's as easy as getting in contact with us. 16S primers are made of forward and reverse sequences, or just a forward sequence, and it's easy for us to add them to the list of available primers. So I would say get in contact with us and we'll do whatever's in our hands to add more primers.
Okay, thank you very much. And a last question about the funding. I think it was not very clear, but how can people buy credits? How does it work exactly? And how do credits translate into analysis?
So on the funding and credit screen, there's that form where users can just write to us a message saying, hey, I have this analysis, I would need this amount of credits. Flomics Stratus support team would get in contact with that user, and we support several means of payment for credits. And basically credits so far translate to a credit per sample. So if you have a cohort of 20 samples that you'd like to analyze with one of our pipelines, you would need 20 credits, and it's as easy as writing to us, asking, and we would provide the voucher code to obtain those credits.
Okay, thank you very much. And looking at the slide that we are sharing now, I see that we have here a voucher code that includes 10 credits. If I'm not mistaken, it means that people can now register in the platform, redeem the code FlomicsDemo10, and they will get 10 free credits to try any of the pipelines that we have available with their own data. And if they have questions, they can get in touch with us. Is that correct?
Absolutely.
Yes.
Okay, so thank you very much. Thank you, Cristina and Julian, for giving us this webinar. Thank you very much to everybody that attended. I think the take-home message is that we can enable your bioinformatics in a very simple and automated way. So if you have any need for automatizing your bioinformatic analysis, feel free to try Stratus with our free credits, or directly get in touch with us and we'll find a way to make it easy and to make it working for you and for your team. So with this, I say goodbye and see you in 2 days for the webinar of genomics services, where everybody is also welcome to join. Thank you very much for the Barcelona Health Innovation Week organization and BioCat for making this possible. Bye-bye. Thank you.
Thank you. Bye.